Here's problem set number 8. We're still looking at X-linked traits. So again, just a quick reminder. XX means that we are talking about biological female. XY means we are talking about biological male. Now, these are on chromosomes, so we're talking about on the X chromosome. So we're looking at how these X chromosomes can be combined with other X's or with Y's to give us different phenotypes. Excuse me. So here we're going to look at hemophilia again. Now, hemophilia is a blood clotting disorder where you're missing a protein required for blood clotting. So you don't make scabs like you should. You just continue to bleed. So it can be kind of dangerous. So we're going to study this. It is found on the X chromosome. Okay, that means that if we are looking at hemophilia as a trait, we have chromosomes that are X. We'll use a capital H for being normal, or we can X with a little h for being hemophiliac. Those are the options, a capital or a lowercase. Or we could have the Y chromosome. Okay, so now that I have my chromosomes, I can look at my different genotypes. And for that, we're just going to distribute. I can put this with itself, with this, or with this. I can look at this one and I can put it with itself. It's already been combined here, or I can combine it with a Y. And Y with anything else without having an X doesn't work. So having YY, that one doesn't work. It won't create light. So we don't have to worry about that one. Now, if I look at these, I can determine my phenotypes also. Move this up a little bit. So I can look at this one, and I know that an X and an X means female, and this one is normal. This one has one of each. That means female carrier. Now this is an X and a Y, so this one is a male, and it has the capital. That means it is normal. Here I have two X's, so I have female, and this one has hemophilia. Here I have an X and a Y, so that's male, but with the little h means hemophilia. Okay, so these are my chromosomes. These are my different genotypes I can get. And it's kind of important to remember that we have the capital H is dominant to the lowercase h. So that means that having normal blood clotting is dominant to hemophilia. Okay, so let's look at here. Show our possible chromosomes. So that's these here. Show the possible genotypes and phenotypes for this trait. Well, that's there. Show a cross between a normal male and carrier female, giving the expected genotypic and phenotypic ratios of the offspring. All right, we can do that. Move it up a little bit so we have lots of space. So we have a normal male. That means XY. And normal meaning it has the capital. And we're going to cross that with a female, so an XX, that is a carrier. Now remember that carriers do not have it but they carry one hiding that they can give. So that means one capital and one lowercase. So we're going to do that cross. I put my different gametes. So here's one gamete, here's the other. Here's one gamete and here's the other. And I bring everything back together. When I'm doing my X-linked traits, I do try to make my boxes a little bit larger so that I can write my phenotypes directly in my boxes. It just makes it a little easier for me to keep track of everything. If I look at this box, I have 2 X's. That means it's female. 2 capital letters. This means it is normal. Okay. Next, we have 2 capital letters, so female. And then it has one of each. That means that this female is a carrier. Here we have XY, but it has a capital. XY means male. And the capital means normal. And then here we have XY, which gives me male. And this one is hemophiliac. So for our genotypic and phenotypic ratios, we're going to have 1 to 1 to 1 to 1 for each. Okay. Let's look at problem number 2. Problem number 2 is talking about color blindness. So again, we have an X-linked trait, but this time, instead of hemophilia, we're going to talk about color blindness. Now, there are different versions of color blindness. We have red-green color blindness. We have blue-yellow. We have total color blindness. On the X chromosome, we're going to look at red-green color blindness. That's the most common. Okay? So, color blindness is X-linked, and normal is dominant to colorblind. So make that a little bit easier to see. There we go. So what are our chromosomes? I can have an X with normal vision. I can have an X with colorblind vision, or I can have a Y. For my different genotypes, I can have this. On this. I can have this one is a female that has normal vision. This one is going to be a female that is a carrier. So she has normal vision, but carries a gene that she can give to her children. This one is male. I guess I could be consistent and use my symbols. We'll go symbol male, and it has normal vision. Okay, and that's because it told us that normal is dominant to colorblind. Down here, I have 2 X's. That means female, and they're both little, both recessive. That means colorblind. Here I have an X and a Y. That means male. With a little b, that means colorblind. So these are all the different possibilities. Here are my chromosomes. Here are my genotypes with their corresponding phenotypes. So show all possible chromosomes. We did that. Show all possible genotypes and phenotypes. Did that. Show a cross between a colorblind male and a carrier woman. All right. some room here. So a colorblind male. That means XY. But colorblind means it has to have one of the recessive traits. Cross it with a female, so that's XX, who is a carrier, so that means has normal but could pass on the recessive. We do our cross. Here's a gamete and here's a gamete. So X with a little b and a Y. Here's a gamete and here's a gamete. X with a big B, X with a little b. And then put them back together. Two X's, this is a female, and this has one of each. That makes her a carrier. Two X's is female, but two little b's means they are colorblind. XY means male, big B means normal. XY means male, little b means colorblind. Okay. What would be the genotypic ratio of the offspring? So it's one of these to one of these to one of these to one of these. Four different, one of each. What fraction of the children, so that means all of them, would be colorblind male? So male and colorblind. So here's male and colorblind. So we're looking at E. That would be 1 out of 4 are colorblind and male. What fraction of the children would be carrier females? So female and carrier, that is again 1/4. What fraction would be carrier males? That's a trick one. To be a carrier, you have to have one of each. Can a male have one of each? Zero or 0 out of 4. You can't have a carrier male. Number 3. For number 3, this one was a little bit harder. I helped you guys start it off in class and get it set up, so hopefully you were able to work this. But let's see if we can find our answers. So, we have a woman who carries hemophilia on one X. So she carries the gene for hemophilia on one X. We know she has to have 2. So on one X, she carries hemophilia. That means she has to be normal on the other one. She carries color blindness on the other X. So that means color blindness on this is this X. That means this one must be normal. So they're opposite. Marries a man who is normal for both traits. Now, men only have one X, and he's normal for both. That means capitals for both. So he is a capital H and a capital B. So we're crossing them and looking at the children. We want the genotypic and phenotypic ratios. So this is just like we've done before. The only thing is our X, instead of having one letter on it, our X actually has 2 different letters on it. And that's not so bad. So we're going to use some color to just help make it clear. All of this stays together. All of this stays together. This and that. so you can see where things are going as I draw. So let's do our cross. I take this gamete and I drop it here. Okay, so here's my blue. I take this one and it all stays together because it's stuck to that X. And I move that over here. My next one moves here. And lastly, our Y. So everything stays together. It's just I have 2 letters on the X instead of 1. It works just like we've done in the past. Okay, but they're stuck together. So those H's and B's are stuck together. So now if I look at the offspring inside my chart, I have an X and an X. This means female. So I can look at the first one, which is hemophilia, and she is a carrier. Then I can look at my B's for color blindness, and she is normal. I can look at the next one, XY. That means this is male. I can look at vision, okay, and this is normal. Oh, nope, I'm sorry. I'm not looking at vision. I'm looking at blood and hemophilia. This one is hemophilia because it has the little H. But for vision, it is normal. Here I have 2 X's, so that's female. I can look at hemophilia. They're both big, so that's normal. Then I can look at vision, and there's 2 lowercase. That means that this one is colorblind. Then I can look here, an X and a Y. That means this one is male. I have hemophilia state is normal. No hemophilia. And the little lowercase b there, though, does tell me that this one is colorblind. Okay, so we just look at them independently. They all stay together, they go together, they move together, and then we look at individual traits. So male or female, hemophilia or no hemophilia, colorblind or not colorblind. Okay, now let's look at number 4. A man, move it down so you can see it too. A man who is a bleeder, that means has hemophilia. That's what they used to call it a long time ago, bleeder. But has normal vision. Okay, so a man who has hemophilia but normal vision marries a woman who is heterozygous. So let's see, a woman who is heterozygous for each trait and recessive alleles are on the same chromosome. So that means my dominant ones are together and my recessive ones are together. Okay, now we can show the cross of these. Again, here is my gametes. So I'm going to move these over here and these on top. and bring my boxes together. And then I can look at the boxes, X and X, that means female. This does not have hemophilia, but is a carrier and has normal vision. This one, XX, is female. 2 little H's. That means this one has hemophilia. A big B and a little b, so this is a carrier for color blindness. You can look here. This one is male. It has no hemophilia, so normal, and no color blindness, so normal. I can see here that this again is a male, but this one has hemophilia and is colorblind. Yikes. Okay, so it's 1 to 1 to 1 to 1. Now, questions number 5 to 7 are all going to use the same information. So we're going to set all that up, and then we'll work through our different questions. Here we go. In tribbles, fur color is a trait. If you're not sure what a tribble is, you can Google it. It's a creature. This trait is X-linked and found on the X chromosome, but not the Y. B is an incomplete dominant allele that causes fur color to be brown. So first I know that B equals brown. Okay, little b is white. And I know that they are incomplete dominants. And the heterozygote one will be tan. Okay, so that's what I know about the trait, but it's on an X. So I have to set this up. We'll start with chromosomes. Okay, so I can have an X with a big B, an X with a little b, or a Y. For my genotypes, I can do my big B with all my different ones. I can do my little b. And that's it. Now for phenotypes, I can have female brown, female, now it told me that the heterozygote one is tan. I can have male brown, I can have female white, and I can have male white. But notice no male tan. That's because you have to be heterozygous. Only this one can be tan. Now, what are our questions? Let's see here. So number 5, write all the possible genotypes and phenotypes. Male and female is important. We got that. Number 6, cross a heterozygous female with a white-furred male. Okay. Move it up so we have some space. So female, that's heterozygous with a white male. So male and white. We fill our boxes back in. One, it's got two X's, so that means it is female. And the color, it's heterozygous, so it's going to be tan. Number 2, this one is male, and it has only a capital B, so that means brown. Number 3, 2 X's means female. 2 little b's mean white. And number 4, an X and a Y means male. And 1 little b means white. What fraction of the offspring would be male? Well, let's see. That's male, that's male, that's female, that's female. So the fraction of the offspring would be male are 2 out of 4, or 1/2. What fraction of the offspring would be brown? Okay, so this one, we have tan, brown, white, white. So we only had 1 out of the 4. What fraction would be tan males? We already talked about tan. You have to be heterozygous. Males can't be. So that's 0 or 0 out of 4. Now, if 12 baby trebles were born in the litter, so in this cross, if I had 12, I would expect them to be divided evenly. So I have 4 boxes. 12 divided by 4 means I would have 3 in each box. Whoops. That means I'd have 3 of these, 3 of these, 3, and 3. How many would be tan female? Well, female tan, 3. How many should be white female? 3. How many should be brown male? Male, brown, 3. How many should be tan male? We have no tan males. That's it. That's the end of problem set 8. If you have questions with these, if you're struggling with any of them, make sure to see me during office hours so I can help you out. Bye, everyone.